In today's video, we're looking at hormones and their receptor pathways. Now, basically, we have two types of hormones. We have the peptide hormone and the steroid hormone. The peptide hormone are water soluble. For example, you have your oxytocin, insulin, follicle stimulating hormone, the LH, growth hormone, glucagon, PTH, and ADH. Then for steroid hormone, which are lipid soluble, we have your thyroxine, which is technically not a steroid hormone, but it acts the same way. We have vitamin D, testosterone, estrogen, progesterone, aldosterone, cortisol, and gonadocorticoids. Now, peptide hormones being water soluble can't go into the cell to carry that function, so they need the receptor outside the cell to help them carry that function. Now, usually these receptors are called G-protein coupled receptors because they have G-protein coupled to them, or they're called 7-pass receptors or sabetine receptors. In this video, we're using the GQ and GS to see the different pathway of peptide hormones. Now, this is epinephrine, for example. Epinephrine stimulates this GS protein receptor. So the GS protein is then turned on by GTP, and the GDP really attached to it is then displaced. So this turned on GS protein goes over to adenylate cyclase, which has a GTPase. And GTPase breaks down GTP into GDP and inorganic phosphate. The energy released is used to turn ATP into cyclic AMP. Cyclic AMP then stimulates a protein kinase A. This protein kinase A then phosphorylates different proteins to carry out this function. It can be to turn on or to open a ion channel, or to move proteins or other things. So, okay, now for the GQ. Now, in this place, what is oxytocin? Oxytocin stimulates the receptor, which causes GTP to turn on this GQ protein using GTP. Now, this GQ protein goes over to phospholipase C, which does the same work as adenine cyclase did here. So, it will have a GTPase, which will break down GTP into GDP and inorganic phosphates. The energy released, phospholipase C, is to break down PIP2. Now, PIP2 breaks down into two compounds, IP3 and DAG. Now, DAG activates a protein kinase C, which does the same work as protein kinase A, so it will start phosphorylating different proteins. Now, also, this IP3, it goes to the smoothie arrow, or the hypoplasmic reticulum if you're talking about muscle cells. Now, once it goes there, it causes the release of calcium. The calcium increases in the cytoplasm, which then goes and binds with camodulin, forming a calcium camodulin complex. Now, camodulin, so a calcium camodulin complex then goes to activate different kinases, which is there to phosphorylate different proteins to carry out the function. Now, oxytocin is very, very important for contraction during childbirth. This site works by increasing calcium in the cell, causing contraction to take place. Now, if we've been attention so far, we might have seen this PED twice, here and also in this part. Now, this PED stands for phosphodiesterase. What it does is it helps us regulate the action of these peptide hormones. Now, PED, it breaks down cyclic AMP and also breaks down phospholipase C. For the steroid hormone, for example, testosterone, they can easily diffuse through the cell. Now, once they diffuse through the cell, they go to the receptor inside the cell or in the nucleus. Now, when they bind to the receptor, they displace this heat shock protein. The receptor goes to a particular gene in the DNA. Now, this gene sequence that it binds to is called the hormone response element. Now, this gene determines the action that is going to take place, whether to proliferate the cell, increase mitosis, or other stuff.